All I really want you to know is the basics of the triglyceride. So I want you to know the structure. It has 3 fatty acid tails and then a glycerol. And these are the tails right here. The bonds that you find in lipids are ester bonds. I typically don't ask that on the test, but you got, these are the tails, this is the glycerol. A lot of times they'll call the glycerol backbone.
You said 3 fatty
Yep.
acids?
Think try, you're gonna have 3 fatty acid tails and a glycerol. Now remember the phospholipid. Phospholipid changes a little bit. You only have 2 fatty acid tails, you have a glycerol, but then you have that phosphate head. And remember that phosphate head is charged, making it polar or hydrophilic, whereas the glycerol and the tails are non-polar hydrophobic. So this is actually one good thing that they did in the book, and it's funny because I think these pictures are way better than some of the other pictures we've ever had. But the micro book has changed with the other group. So I work on it, 2 different books, and it kind of is what it is.
Wait a minute. On the phospholipids, it has a fatty acid tail, right?
2 of them.
2 of them, okay. So that one,
So 1, 2, and a glycerol and a phosphate.
okay, I wrote it in the wrong spot.
I should have brought water. That heat's drying me out. I don't know if I'm sitting. Right.
There's the one.
No, I feel it's like blowing all on me. So remember the cell membrane's made up of phospholipids. So it's a bilayer. So there's 2 rows of them. So those 2 fatty acid tails, they point towards each other. And those phosphate heads, because they're hydrophilic or polar, will point towards the inside and outside of the cell. Now, if you were to drop phospholipids in a water, one single layer, it forms a circle. So the tails still stay together. The heads interact with water. That's how that works. Cholesterols are example of steroids. Remember, steroids have ring-like structures. So where you see all those rings in the structure, that's typical, very typical of a steroid. Cholesterol is the example I want you to know. And that is also part of the cell membrane, and it helps to stabilize the cell membrane, makes it a little more firm. Remember that membrane is very flowy. It does help to make it a little more firm. And those are the 3 types of lipids. Now proteins, those are most definitely the predominant organic molecule you find in cells, and talk about proteins quite a bit. What are the building blocks of proteins?
Amino acids.
Amino acids, and they are linked together with what?
Hydrogen.
Peptide bonds.
Oops.
But you find hydrogen bonds within that structure. And I might even, I probably have a picture, thought I did, of the hydrogen bonding. But as far as the protein, peptide bonds in between amino acids. Now, some examples there, enzymes. Enzymes are great examples of proteins that they can change shape. Sometimes their binding site's open, sometimes not. Antibodies are also great examples of proteins. And even though we've been born with the ability to respond to any antigen, sometimes there's a need for tighter binding. So that actual binding site can shift around just a little bit so that it can increase the tightness of the binding to the antigen itself. So it's also going to shift around theoretically a little bit. So proteins are nice in the fact that they can move. There's a lot of other protein examples there, but I'll leave it at that. If you take a look at this picture, you'll see some of the different amino acids. Now there is a part that's always the same and a part that's different. That R group is the different part. So not in your book, I think they still do about the same thing with this. It's a little different color for each shaded area that's different. So if you take a look, the part that, the H's and the N's and the C's and the O's that are in blue, that's all the same. But when you get to these different colored blocks right here, this is the part that changes between each amino acid. So it makes it what it is. So that would differentiate valine from phenylalanine, for instance. You do not have to know the 20 amino acids. Shoot. You're dying to do that, I know. I remember in biochem we had to draw all of the amino acids. That was part of the test. It was just ridiculous. I would just sit at home and just draw all day long, all night long. See, your hand just did it. Now, one of the things that I ask you on your exam is descriptions of all of these different macromolecules. So, as far as the structure and where the bonds form, when we talk about amino acid bound to amino acid, there's a part to the amino acid that's the amino end, and there's another part that's called the carboxyl end. And that's all in the top there, the wording. Now, these little dashes here are the peptide bond. So from amino acid to amino acid, the carboxyl end, there'll be a peptide bond between it and the next amino end. Now, these little R's are indicating that's amino acid 1, that's 2, that's 3, that's 4, that's its R group. That's what makes it what it is. Now, in the second amino acid, again, that carboxyl end where you find the C, there'll be a bond that forms between the amino end. And it goes all the way down the line like that. So how amino acids are put together, amino acid, peptide bond, amino acid, that bond is from the carboxyl end of the first one to the amino end of the next one. Carboxyl end of the first one to the amino end of the next one. Now I'm sure you guys remember, oh well, this shows you a lot of the hydrogen bonding in here. See all these dashed lines? That shows you all the hydrogen bonding in the proteins. You guys remember the 4 different levels of structure to a protein? The primary, just pretend that it's being built on that ribosome and the amino acids being linked together, peptide bond, amino acid. So it's just in a string. When you get to the secondary structure, because proteins have to form shapes and fold, the second structure includes the alpha helices and the beta pleated sheets. So you got coils and pleats. So the alpha helix is the coil, the beta pleated sheet, those are the pleats. So the secondary structure starts to make those different shapes. The tertiary structure still has all those shapes and they'll indicate the coils and the sheets, but it's starting to fold on itself. Now the quaternary structure still has the beta pleated sheet, the alpha helices. It is folded, but it's multiple polypeptides put together. And those polypeptides will all have a 3-dimensional shape. So this one includes these 2. This right here is as basic as it comes. So think of it being built. It'll come off as a string. And then it'll start making these shapes. Then it'll start folding after it made those shapes. Then multiple chains come together folded with those shapes in them to be that complete protein. So on the exam, I might ask you a description of each of those levels. Now remember, in this structure, it can move around and change shapes. Sometimes enzymes have multiple binding sites where they may be exposed at certain times, then another one's exposed at another time, and so forth. So it's got to be able to move around. So hydrogen bonding is present through that whole thing. And the last macromolecule that we have, is any questions about proteins? I'm sorry. Don't want to go too fast there. Any other questions?
Can we pick your brain? Like you and Dr. Meisner, I would love to just listen.
There's not much in there besides this stuff. I don't really have any room for any more.
I guess not.
Flash drive.
Yeah.
That would be awesome. Or
Yeah,
just
that
stick
would.
this in your ear, download it.
What are those phones, you know, where you can touch them and they?
Yeah.
That'd be cool.
New definition of high five.
Christine.
That'd be awesome.
Yes, it would.
That would be awesome. Well, I do that to a lot of people. I don't know that it all fit though. I don't know, might have killed some brain cells here and there.
Yeah, we don't want to go there.
I don't know how many I got living.
We don't want the PhD level, we just want.
We just need we don't need the Velcro, right?
Yeah, we don't need all that, we just need like the basic.
Oh, okay. You guys remember your two nucleic acids, deoxyribonucleic acid and ribonucleic acid. Know the long way written out, what DNA stands for and what RNA stands for. Remember DNA is double-stranded, RNA is single-stranded. That DNA contains all the genetic information. Remember, through the process of transcription, transfers it to the RNA, and then in the process of translation, that RNA will translate it into a protein. Now, you guys remember some of the other differences between RNA and DNA,
The adenine
right? Well,
at the golf course
close.
and at your golf course.
remember DNA has thymine.
That's how I remember it.
GTA, and then the U changes on the
AUGC.
RNA has the U.
Right, the RNA has the U, the DNA has the T.
Hey, I remember.
Both have G, C, and A. G, C, and A is present in both DNA and RNA. It's just DNA just has the T, RNA just has the U. So that's one other difference. What about, you guys remember the building blocks of DNA and RNA, right? They are called
Nucleotide?
nucleotides. So you could say it is a polynucleotide chain, and DNA is a double polynucleotide chain. So the nucleotide is the building block, and at the top it shows you the three parts. There's the nitrogen base, which is either the adenine, guanine, cytosine, thymine, or an RNA, a uracil. Now it's got a phosphate group, and the pentose sugar, what's the difference there?
That's 5C sugar. 5 sugar.
Right, it is a 5-carbon sugar, so it's a pentose sugar. It's got 5 carbons instead of 6. I know, I hate to shut the door because it's gonna burn us alive, but I'm glad the heat went off for a second. Remember, the sugar's ribose in RNA and deoxyribose in DNA. So make sure you know those three differences there. DNA versus RNA, double strand versus single strand. The sugar, deoxyribose, ribose. You have thymine in DNA, but you have uracil in the place of in RNA. Is all that stuff coming back?
Mhm.
Slowly but surely it is. Now, you guys remember the A always matches up with the T. So now if you take a look at this picture right here, this is the sugar, the phosphate. So this right here, they consider these the backbone. That's the backbones of DNA. RNA is the same thing, it's just got one side. So the sugar and the phosphate, that's the backbone. The bases are connected by hydrogen bonding, which is a good place for the weakest bond to be there, because remember, DNA has to open up to replicate itself, to make RNA, all that good stuff. So if it's making proteins, it's gonna open up quite frequently. Make proteins more than it'll replicate, but. So sugar, phosphate, that makes the backbone. And you can see that the lines here are indicating that it is a stronger bond here. So this is not meant to break apart, whereas this is. So if you take a look, the A's always go with the T's, the G's always go with the C's. So if I ask you on the exam for a complementary sequence to one side of DNA, so with this one, you just, if it's an A, there's a
G.
C, there's a G, there's a T, there's a, okay. So you guys could do that, that's your easy question. Now, if I say during the process of transcription, you guys need to know you're making RNA, can you give me the complementary sequence that would be made in RNA? So this would be, if there was an A in RNA, there would be a
U.
U. If there was a C in DNA, there would be a G. If there was a G, there would be a C. There was a T in DNA, the other side of RNA would be U.
That's that in.
So I just passed.
Oh, hey.
Oh, that's a good for Doug.
I didn't know for sure, so I didn't know she was here. Sorry.
That's okay.
I'm passing the micro book. I had to give it back to Doug. Doug gave it to her.
There you go, that's a better way to get it.
Okay.
So if you had a T in DNA, what would be on the RNA?
Be U.
An A. Remember, there's plenty of A's in RNA, just not U's, so that's the little trick there.
So
There's
how
a
do
T
you
in
figure it out?
DNA, an A will match up. Do what?
How do you figure this out?
The A's always go with the T's, that's a rule. The G's always go with the C's. If I'm asking for replication, I want that DNA side. If I'm asking for transcription, we're making RNA. So you could actually, if we're making RNA in the process of transcription, the rules still apply. The G's always go with the C's, but the A's go with the U's.
And that little phrase works at the golf course and at your golf course.
Well, yeah, so you would just do that U instead. But remember, and that's why I always like to do this, remember if there's a T in DNA and you're making RNA, there will always be plenty of A's in RNA. There you
Say
go.
that one more time.
There will always be plenty of A's in RNA.
Say
So
it
if there's a T in DNA,
Right.
the other side will be an A in RNA. But if there's an A in DNA, and you're making RNA, it'll be a U. So really, if you write the complementary strand down to that side of DNA, and you're making RNA, you just sub in U's for those T's. Do you guys want to do a practice one?
Yeah.
Yeah, that would
Yes.
hurt.
I think we're like deer in the headlights.
Okay. Do, all right, write T-A-G-C down. T-A-G-C.
Okay.
All right, give me the DNA. Give me the other side if it's DNA. T-A-G-C, what would that be? A-T-C-G.
Oh.
And that's complementary, right?
Right, that would be the complementary side of DNA. Right, write that sequence down again, T-A-G-C. Now in transcription, what would the RNA be to that sequence? A-U. CG.
Yeah, see a light bulb.
No, see, that's where.
That's just
I
a
get
little
mixed up on the
review.
RNA. Do I take? I'm getting mixed up. Get lost?
So TAGC, if we're making RNA right alongside, and we know it's DNA because it's got a T in it. So if we're making RNA right alongside of it, the T would order an A there for RNA. The A would order a U because there's no T's in RNA. The G would order a C,
See,
yeah, and then it's released, but
okay.
so it's really the same thing as what DNA would be, but no T's, just U's in it.
Okay. I remember now.
Everybody clear on that?
So the first example that you gave us about was.
DNA and DNA, right. Mhm. And the second one was transcription. Okay. Right, are we okay with the structures here?
After we practice it.
Yeah, and just write yourself a couple of sequences down. And if you want to write some sequences down and practice them over the weekend, we can talk about them and check them on Monday.
Okay.
So if you guys want to make a good one and put them up, we'll put them up on the board. We got a board that'll stick out a little bit from just the PowerPoint. We'll do some of those.
Okay, thanks.
Now, on this slide, there's one of these terms I want you to know. At the top, the term nucleoside. That's not quite a complete nucleotide. The nucleotide has, remember, the base, the sugar, and the phosphate. The nucleoside only has the sugar and the base. So the nucleoside is the nucleotide minus the phosphate. So nucleoside means it's just the sugar and the nitrogen base. Now these are the 2-pentose sugars. Remember, deoxyribose is in DNA, ribose is in RNA. Now, the next thing we got to go through is these nitrogenous bases have classifications. They're either a purine or a pyrimidine. And I think your book still has the same, is it the same picture? And it's a good one to start there. Yeah, it's a little different. Actually, this is a little bit better than the other one. The purines have double ring structures, and the A and the G are classified as a purine. The pyrimidines have single ring structures, and the T, the C, and the U are all pyrimidines. So there's more pyrimidines than there are purines. Purine, double ring structure. Pyrimidine, single ring structure. You guys will need to memorize which one's a purine and which one's a pyrimidine. Yeah, I do like that picture just a little bit better. One of the things I want you to notice, and this is the same picture here, right at the bottom. If you take a look, there's different amounts of hydrogen bonds between the A and the T, 2, and then there's 3 between the G and the C. Don't know that I'll necessarily ask that, but I do want you to notice that there's different amounts of hydrogen bonds. 1, 2, 3 between this guy and 1, 2. But now one of the things that you'll notice is that you always have a double ring structure, and on the other side is a single ring structure. So a purine always hydrogen binds to a pyrimidine. Does everybody see that in that picture? If you look on the other side, it's always a double ring and a single ring. And the last thing that I want to bring up, you guys remember, and you guys should have this picture from A&P, that DNA is anti-parallel. And I'll bring this picture up. You can find it on the web, but let's see. Now You guys still remember this? One side runs 5 to 3, the other runs 3 to 5.
Yeah.
So if you take a look at it, it's anti-parallel. You look at the bases in the middle that are hydrogen bound. If you took this sugar and phosphate and just flipped it over the other way, that's the anti-parallel. So this, and these are all where the points are, the carbon positions, except the fifth one is up here on the little leg. But you see the point, see how it's flipped over on the other side. So now why it says it runs 5 to 3 and the other side is 3 to 5. That sugar has 5 carbons on it. So that's telling you that the phosphates connect to the sugar, to the phosphates, to the sugar at the 5th and 3rd bonds, or the 5th and 3rd carbons. So that's where that bond is forming. So again, this first point's the first carbon, 2nd, 3rd, 4th, and on the little leg is the 5th. And you'll notice, always between the 5th carbon and the 3rd carbon, there'll be a bond. And it doesn't matter what side it's on, it's just flipped over the opposite way. So the 5 to 3, 3 to 5 is how the phosphates and sugars connect together. How the phosphates and sugars connect together. And the numbers represent the carbon position on the sugar. So at the carbons where a bond's going to form. Nothing happens at the 1st, 2nd, or 4th carbon. Just at the 3rd and the 5th.
Where did you say we can put it?
It's on the web. I think I probably put into Google like anti-parallel DNA and it popped up. And there's tons of other pictures too. And it probably wouldn't be a bad idea to look at several resources. I'm sure I got it from some website that had a lot of DNA pictures and stuff. But I like this one because they actually labeled it up here at the top. You'll never have to draw this or label it. I just want you to know what that anti-parallel means. And those numbers 3 and 5, that's representing carbon position on the pentose sugar. And bonds form between the phosphates and sugars there. I'm not as concerned at where it's bonding to the basis for that anti-parallel term. Don't know that I will ask you a question this time. Sometimes I save it for the DNA chapter, so. But it's just a description. And that's all.